Hello everyone, welcome to today's session. Today we are going to see Class 12 Biology, Chapter 7, Human Health and Disease. In that we are going to see cancer. So let me start with a simple question. So what happens when the cells in our body divide? Yes, very good. So new cells are produced. So cell division is actually very important. See, our body needs new cells for growth, repair and replacement of old cells. But what happens if cells keep dividing without control? See, that is where the problem begins. So, cancer is a disease in which the normal control over the cell growth and differentiation breaks down. So, what is cancer? Cancer is a disease in which the normal control over cell growth and differentiation breaks down. So, normally, our body has a very good control over cell division. But in cancer cell, this regulation is lost. So, next we are going to see the contact inhibition. Contact inhibition, this is the body stop signal. So what happens in contact inhibition? See, this is a very important NCERT concept, children. You have to understand this. Yeah, this is very easy also. Just imagine you people are sitting in a classroom. Okay. And if there is enough space means everyone can sit comfortably, right? But once all the seats are occupied, nobody else can keep sitting in the same place, right? So the same way our normal cells behave somewhat similarly. So when we take this normal cells, when they come into contact with the neighboring cells, their growth is inhibited. What happens? When normal cells come into contact with the neighboring cells, the growth is inhibited. And this property we call as the contact inhibition. This is the contact inhibition. So contact with other cells stops the growth of the normal cells. But what happens in cancerous cells? See, cancer cells lose contact inhibition. Yeah. So no proper stop signal will be there and the continuous cell division will occur. And because of this, what happens? Accumulation of the cells takes place and it results in the tumor. So this diagram you can able to see, this is the normal cell. Yeah. So it stops at contact. But here what happens, it has lost its contact inhibition. So what happens, it piles up uncontrollably. So this leads to the formation of the tumor. So students, if normal cells have a stop signal, and what happens when cancer cells lose the signal? Yeah, they continue dividing uncontrollably. Then what is a tumor? See, when cancerous cells continue dividing, they can form a mass of cells called tumor. We have two types of tumor. One is the benign tumor and another one is the malignant tumor. So in benign tumor, let us see this by differentiation. Okay. So let us take the growth. When we talk about the growth, growth in benign tumor is usually it is localized in one place. But in malignant, it is a rapid. Yeah, it grows rapidly. And when we talk about the location in benign, it remains at original site. But what happens in malignant? Yeah, it can spread. Yes. And then when we talk about the invasion, see here in the benign, it does not invade the surrounding tissue. But here in malignant, it invades the surrounding tissue. And when we talk about the damage, so usually it causes little damage, but here it can cause considerable damage. It causes considerable damage. And also how it is spreading. See that this does not spread to distant sites, but they can spread to distant sites. And we talk about some important term over here. It is confined. Yeah, it is confined. And this is metastasis. Yeah. So just how to remember this? See, think of a benign tumor as staying in its own neighborhood. And when we talk about a malignant tumor, it does not stay there. It can invade surrounding tissues and spread to distant parts of the body. Then what is this metastasis? See, this is the most feared property. This is the most feared property. So what happens here? See, suppose a malignant tumor is present at one location. See, some tumor cells can break away, right? They break off, yeah, they enter the bloodstream and they travel and they reach another location and there they form a new tumor. See, this spreading of the cancer cells to distant sites is called as metastasis. Yeah, the spreading of the cancer cells to the distant sites is called as metastasis. So just imagine a tumor is present at location A. Okay, so if its cell travel through the bloodstream and establish another tumor at the location B, and what is this process is called as? Can you tell me? Yes, very good. It is metastasis. So next we are going to see the causes of the cancer. So we have a question, right? Why do normal cells become cancerous? Yeah. See, several physical, chemical, and biological agents can cause cancer, and these cancer causing agents are called as carcinogens. These cancer causing agents are known as carcinogens. And let's divide them into three groups. So one is the physical agents and chemical agents and biological agents. So let us start by the physical agents. So physical agents, so we have physical into two again, is the ionizing radiation and non-ionizing radiation. When we talk about the ionizing radiation, yeah, it is x-rays and gamma rays, so they damage the DNA, yeah, and also leading to neoplastic transformation. When you talk about the non-ionizing radiation, so UV rays, they also damage the DNA and they trigger the cancer. Okay, this chemical agents, what are they? See, chemical agents are present in tobacco smokes, yeah, tobacco smoke, and these have been identified as the major cause of lung cancer, yeah. And then when we talk about the biological agents, so some viruses can cause cancer, and these are called as oncogenic viruses, and they contain the genes called viral oncogenes. They contain genes called viral oncogenes. Then what are proto-oncogenes and cellular oncogenes? Yeah. See, this sounds complicated, but let's make it simple. Okay, so our normal cells also contain certain genes called as cellular oncogenes or proto-oncogenes. So under certain condition, when these genes become activated, they can contribute to oncogenic transformation. Okay, so under certain conditions, what happens when these genes when they become activated, they contribute they contribute to oncogenic transformation. Okay, always remember viral oncogenes, they are associated with cancer causing viruses. Okay, and then next we are going to see cancer detection and diagnosis. So just now imagine someone has suspected a tumor. So how do doctors find out whether it is cancer? See, early detection is extremely important because many cancers can be treated successfully when detected early. So, the first one we are going to see is the biopsy. So, when you talk about the biopsy, a small piece of suspected tissue is taken. Okay, a small piece of suspected tissue is taken and is then cut into thin sections. Then it is stained and it is observed under the microscope. See, this microscopic examination is called as histopathological studies. Okay, so this examination is called as the histopathological study, and a pathologist examines the tissue. Okay, so next we are going to see the bone and bone marrow test. So these are particularly useful in detecting the increased cell counts in leukemias. Okay, so next one is the radiography. So it uses X-rays to detect the cancers, and particularly involving the internal organs. And next one is the CT scan. CT scan name is computed tomography. Okay, it uses X-rays. It uses X-rays to build a 3D image, or we can say it's a three-dimensional image of internal structures. Okay, then what is MRI? See, MRI is magnetic resonance imaging. It uses strong magnetic fields and non-ionizing radiation to detect the changes in living tissues. Okay, here's a simple memory trick for detection. Okay, so let us see. So just a ma'am is saying BBXE ma'am, BBXE ma'am. So B is for a biopsy, and another B is for a blood or bone marrow test, and X is for X-ray, C is for CT, and M is MRI, and A is antibody test, and M is molecular biology techniques. So next we are going to see antibody and molecular test. See, some cancer cells, they have cancer specific antigens. Yeah, they have cancer specific antigens. So these antibodies against these antigens can help to detect certain cancers. Okay, so antibodies produced against these. Okay, so what happens? It helps to detect the certain cancers. And molecular biology techniques also, it can identify the genes which are associated with inherited susceptibility to certain cancers. Okay, and why is this useful, children? See, because identifying such genes may help in prevention. Yeah. So next we are going to see the treatment of cancer. So this is the final major section. You should know how is cancer treated. So the major approaches include the surgery, first one. See, the tumor mass is physically removed. In surgery, what we'll do? The tumor mass is physically removed. And in radiotherapy, what we'll do in radiotherapy? See, radiation is used to destroy the tumor cells, while we take care to protect the surrounding normal tissues. So always it is a concern, right? We should protect the normal tissues which are surrounding the tumor. And next one is the chemotherapy. So in the chemotherapy, certain drugs are used to kill the cancerous cells. So some drugs are specific to particular tumors, and chemotherapy can have side effects also. So some are like hair loss, anemia, yeah, and immunotherapy. So cancer cells can sometimes avoid detection and destruction by the immune system, and use substances called biological response modifiers may be given there. Yeah. So this is a very important example. And I mean, and the alpha interferon. Yeah, what is this alpha interferon? See, it helps to activate the immune system. What it does, it helps to activate the immune system and assist in destroying the tumor cells. Yeah, immunotherapy, right, children? And then when we talk about the combined approach, why we need a combination treatment? See, this is a very important thing. And why don't doctors just use one treatment? Why they are opting for so many? See, the answer is that different treatments work in different ways, that we know very well. But many cancers are treated using a combination of surgery and also radiotherapy and chemotherapy, and immunotherapy may also be used here. So we have come to the conclusion part. So let's put this entire cancer thing into a story. Okay. So normally our cells divide in a controlled manner. Yeah. And they also show contact inhibition, which helps to prevent the unnecessary growth. But when the regulatory mechanism breaks down, see, the cells may start dividing uncontrollably, and these abnormal cells can form a tumor. See, and if the tumor remains confined to its original location, it is generally called as the benign tumor. But if the tumor grows rapidly and also invades the surrounding tissues and can spread to distant location, it is called as the malignant tumor. And the spread of the malignant cells to distant sites is called as metastasis. And cancer can be triggered by physical, chemical, or biological carcinogens. And also early detection can involve the biopsy, histopathology, bone and bone marrow tests, and also X-ray, CT, MRI, antibody test, and molecular techniques. And finally, cancer can be treated using approaches such as surgery, radiotherapy, chemotherapy, and immunotherapy. So we have come to the conclusion part. Watch the full video, children. Don't skip the video. Yes, we'll see you in the next video. Thank you.